everyone for joining us again today for, I believe, our 7th virtual town hall. I keep numbering these things and then checking the number before we get started. But this has been a really great experience for us. We're really glad that you all continue to join us. And just want to say thank you. Hope everyone is staying healthy and well, right? That's the number one thing. And hopefully, there's been some good news, relatively lately, that people are making progress and innovation is happening. So I think we have a lot to really be optimistic about. We have a really great program today. We've got a great group of speakers from Boston University's National Emerging Infectious Disease Labs and Brainy IRB. Just a reminder, if you have questions, you can use the chat bot or you can use the raise your hand option, and we will call on you. If you're not speaking, just stay muted so we avoid background noise. And just a little update on things that Mass Medic are doing. We've had a couple great webinars in the last week on human factors and on designing products for the home. If you missed either of those, we do have video. So please feel free to reach out to me. We'll be happy to share that with you. Sunrise Labs and Goddard are two of our excellent partners who put those on, and we got a lot of great response. So if you missed them, please reach out and we'd be happy to get you the information. Also, we're starting our brand new Women in MedTech virtual series this evening. So if you have not signed up for our first event tonight at 5 p.m., please feel free to do it. It's not just for women and it's not just for people in MedTech. So it's a really great 4-series program that we're starting tonight. It's going to run for the next 4 weeks, one a week. We have some excellent speakers, some really great representation from local governments. We announced yesterday that Attorney General Maura Healey will be joining us for our last event, and we have an exciting new speaker announcement for our 3rd event as well. Representative Joe Kennedy is going to be making some opening remarks to give us just his take on how to stay motivated and how to make the most of the moment, but as well as how to support local organizations like New Hope, who is our partner on this event. So if you'd like to learn any more, please feel free to send me a note. You can shoot it in the chat bot or email me at rachel@massmedic.com. Otherwise, I'm going to turn this over to Brian to give just a few comments and updates on how things are progressing.
Thanks, Rachel, and thank you everyone for joining us. Week 7 of White Collar Criminal Fantasy Camp. This is what it's like to be under home confinement, and I have to admit, I always thought it was a cushy sentence, but I think I'm changing my mind on that. In all seriousness, Rachel is right. There has been a smattering of some good news over the past couple of weeks in terms of our hospital's ability to withstand the surge. I think there is high confidence now that our hospital systems will be able to withstand this initial wave, and that we, as a community, are meeting the needs of our healthcare partners. It's interesting, I had a conversation a little bit earlier this morning with a group in robotics, and we were talking about how we can, how robotics can get involved in healthcare, and it just dawned on me, we're all involved in healthcare now, no matter what your profession is. We are all involved in the pursuit of healthcare and healthy living here. So there's so many ways to get involved. I think a couple things to pay attention to over this week, just in terms of things from the state. The governor opened up some new funding for senior assisted living facilities. I think we cannot quite comprehend the scale of the tragedy in senior assisted living. I think by the time this is over, we're going to realize that we've witnessed something very profound and very, very tragic in that community. So there's a lot of innovation that needs to happen to help keep our seniors who are living in these communities healthy. There's a lot of opportunity there, and there is new funding for that. So I would pay attention to that. I would pay attention to, on our COVID-19 resource page on massmedic.com, we have updated with some back to work protocols that we worked on with MassBio. These are very sort of rudimentary, but very good, solid tips for how you can start preparing your workforce for bringing people back. And again, you can find that on our resources page. Finally, as Rachel said, we continue to provide a lot of um, content online. We're going to continue to do that. We have officially postponed our annual dinner, which was set for July 8th. We are looking at dates in the fall. We are all very hopeful we can get that off. Again, just one more thing, there's an interoperability webinar coming up in a couple of weeks that we're going to send out some information for that I think you should take a look at. But I'm particularly excited today because I'm excited to get an insight into a little more about the virus of COVID-19, learn a little bit more about what our neighbors are doing. For those of you who have never been to our office, we're at 650 Albany Street. And then right next to us is the building they call the NEIDL. And so I'm really excited to find out what's going on in there. It's a big tower of a building hidden behind some really intimidating looking fences, but they have a beautiful lawn that I've always wanted to run on, especially in the summertime. But I'm really excited for this program. I think the more we can start to learn about what our community is doing, it makes me feel better. So I just want to thank all you. I know how hard everyone's working. I know under what difficult circumstances we're operating under, but you all are doing an incredible job. And the feedback I'm getting from our stakeholders in government has been great. They appreciate everything you're doing. We have their ear now more than ever. We're going to continue to work on using this time as a lessons learned opportunity to figure out how we make a better healthcare system. And we will have a moment over the next year where we have their ear. So let's come up with the best ideas, and let's get our industry not only back to work, but coming out of this better. So Rachel, that's all I have, but thanks for the time.
I think that's perfect, and it's a great introduction to Dr. Ronald Corley, who's working over at the NEIDL. I will turn it over to you at this point. I think this is a great opportunity, and we're so thrilled that you're joining us.
Well, thank you, Rachel and Brian, very much. What I'm going to do is just give you a little insight into what's been going on in the NEIDL since this outbreak really began. And so what I'm going to do is just kind of summarize in a brief way what we're doing, but I'm happy to answer any questions. But I'm going to tell you a little bit about how we got here and what it takes to actually get started on working on a virus such as SARS-CoV-2. So I think we all remember that this started just 4 months ago, really. Right at the end of December, when it was announced that there were some people with undiagnosed pneumonia in China, and that number continued to grow. And within a week, the Chinese laboratory in Wuhan actually presented posted online the type of virus that was causing this disease, and it was defined as a coronavirus. And this is coronavirus is a virus that is relatively circular, as you see. It's got a RNA genome, so it's very much like all of the emerging viruses that anybody deals with. It's an RNA genome. It's got 4 proteins in it that make up the virus, and then it's got a ton of what they call non-structural proteins. Many of them are designed to antagonize a cell's immune response so that when it infects a cell, the cell doesn't recognize it immediately. And this allows it to actually grow within cells. So coronaviruses have been known for a number of years. The first couple that were human coronaviruses were discovered in the 1960s. These viruses infect a large number of mammalian species as well as birds. Then there were the 2 big outbreaks, the SARS-CoV outbreak, the initial one in 2002 and 3, and the MERS outbreak that still goes on in the background in the Middle East. And this particular virus is the 3rd more pathogenic coronavirus, and I suspect it won't be the last one. Now, what caught our attention when this first started is plotted here. Based on WHO data, if you look at the SARS rate of transmission and the number of cases in the first 120 days or so, it flattened out very, very quickly because it could be brought under control by public health measures. The novel coronavirus, as you can see, spiked very, very quickly. And there were a couple of reasons potentially for this. The first was that it was already circulating widely before people recognized it, and those individuals were then spreading it to others. But it was also possible that the transmission of this virus was different than the original SARS. And I think we now know that that's the case. First of all, in the original SARS virus, if you look at viral load in the respiratory tracts, this is how you spread the disease. The virus load doesn't really peak for about 10 days after onset of symptoms. So you can identify people with symptoms and quarantine them and take public health measures to ensure that they're not infecting people with that disease, which is probably why it was brought under control very rapidly. This particular virus, SARS-CoV-2, has high viral load soon after system onset. And in fact, there's now evidence for asymptomatic transmission. Most viruses aren't transmitted asymptomatically. Measles is another one that is. And this is why I think we're having the biggest problem controlling this, is because we don't know that people are transmitting the virus to other individuals before they actually start showing symptoms, if they ever show symptoms. And about half the people that are diagnosed in a large number of studies at the time of diagnosis, in fact, have no symptomology. They may go on to develop it, but this just goes to show that this is a highly transmissible type of virus in terms of transmissibility. So we weren't the only ones that were really interested in this virus. Everybody in the Boston biomedical community really started getting together and talking about how do we actually deal with this virus. Now, one of the things you should know is that there was nobody in the Boston area that worked on coronaviruses when this virus outbreak started, which I think is absolutely fascinating. There were so few people around the world that are actually true coronavirus experts. So everyone had to become expert very, very quickly. And I think through the work of George Daly, the dean at Harvard Med, and Bruce Walker, the head of the Reagan Institute, started talking to a number of people from institutions around here, including people in the NEIDL, saying that in order to do the science that's necessary, that we really ought to begin to get together in order to collaborate effectively and widely in various ways. And so this first meeting happened in early March. Now, everyone was already trying to develop tools. And so for us, the first thing we needed to do was try to get the virus. And I think, what I'm showing here is just to try to give you a little idea of what was going on when we tried to get the virus. So in early February, we had made the commitment that we were going to start work on SARS coronavirus 2 in the NEIDL. And so we submitted the first protocol to allow us to begin to receive the virus in early February. And as you can see at that time, only 15 people in the US were known to be infected with the coronavirus. By the time that we received the virus, that number had gone up to only 60, but we still weren't ready to begin to study the virus at this particular time. So we had just received it by the end of February, and then it took another 2.5 weeks to get all the final approvals to start working with the virus. It's a biosafety level 3 virus. And actually, the groups that could jump on it the fastest in the NEIDL were people that worked in biosafety level 4, and there were various reasons for why they wanted to work in biosafety level 4. None of them have to do with the pathogenicity of the virus, but they have to do with more practical considerations. Those practical considerations are the instrumentation that they needed was already set up in biosafety level 4. And the other thing is we already knew that there was a PPE shortage of healthcare workers. And so in working in biosafety level 4, they don't need to rely on the same PPE that healthcare workers are working on. So By the time I transferred the virus to Rob Davey, which was on March the 19th, we had gone from 15 cases in the US to about 14,000 known cases in the US. So you can get a sense of how concerned we were in trying to get this work underway as quickly as possible, and the entire Boston community was the same. So this is Rob Davey right here. That is the virus in that container that he's taken into his laboratory in the biosafety level 4. He works on therapeutics, so he has high throughput robotics to allow him to work on screening mechanisms. And he is doing screening now for a large number of people around the Boston and greater Massachusetts community, as well as already for a number of companies that have reached out to him to ask him to screen many of their compounds. Now, at the same time we were trying to get science started, we also knew that there were some shortages that we might be able to help with. First was diagnostics. We know how the rollout of that impacted the spread in the United States. Personal protective equipment, particularly N95 respirators, were in short supply and still are in short supply. And everyone was already worrying about serologic assays. These are assays that test for antibodies that allow you to determine who's been exposed. And so a number of groups at a number of institutions started working on all of these. I will give you one example of what we did. We took the COVID Innovation Reuse Task Group from Mass General Hospital, which was already being overwhelmed, even before our own hospital, Boston Medical Center, was. They asked if we could actually test these masks for whether or not they could be decontaminated effectively and screened for virus. And so Anthony Griffiths and our environmental health and safety group worked with this group and worked with V&V Testing, Massachusetts company, and asked the question, can you actually prove that these vapor sterilization, hydrogen peroxide methods actually kill all the COVID-19 viruses that are on N95 respirators? And they successfully did that. And as you know, there are a number of hospitals now, including Boston Medical Center, that's using this methodology that Patel has now rolled out and allowed people to work with. And so that we can repurpose N95 respirators if we need to. And I think everyone has a level of confidence in this. Serologic assays are then something that we and others are also jumping onto. And this is important for a large number of reasons. I know there's a lot out there in the community about, well, this is going to tell you who's already had it, and therefore it's going to tell you who is actually protected. And we don't really know that, in fact. So we don't know whether or not every patient that recovers, including those that have relatively asymptomatic diseases, have high concentrations of antibodies, and whether their age, gender, or racial differences in these antibodies. So this will allow us, and you're hearing all kinds of numbers now about how many asymptomatic infections are out there. And then we need to know, how long do these antibodies last? And is there a correlation with immune protection? In other words, are people that have seroconverted, have made antibodies, are they actually resistant to reinfection? And we're not going to know that, I think, until a number of people who have recovered get re-exposed in the future. And the reason that information, we're all a little bit, non-committal on it, is because when you look at the other coronaviruses, those that cause the common cold and upper respiratory infection, people can be reinfected after a period of time with those. And so it's likely that individuals that recover from this disease will be protected for at least some period of time, but we don't know what that period of time is. And trying to understand that is, I think, really critical to how we not only design vaccines, but also develop strategies for how often individuals are going to have to be re-immunized with these vaccines. So we are now virtually everybody inside the NEIDL has been working on SARS-CoV-2 and the COVID-19 disease. So we have a large number of people that are engaged in developing clinical studies and clinical trials, but also that are setting up to do the preclinical studies that you need for identifying therapeutics and vaccines that may work. And let's just list a few of the types of studies that are being carried out. And then a number of people are also working on the immune responses so that we can really understand more about what this particular virus actually does in terms of generating immune responses, and then maybe use that information to develop better therapeutics as well. So as this disease has spread, clearly there are a large number of other things that happen, and these are all things that need to be researched as well. I mean, there are newly recognized symptoms, you know, other than the fever, cough, and shortness of breath that they talked about early on. There are chills and repeated chills. There are muscle pains and headaches and sore throats, loss of taste and smell, even preceding lung symptoms in some individuals. And it has a very, very different phenotype than it has in patients with other types of viral diseases. Just heard a talk about it yesterday from a BMC doctor. And then there's gastrointestinal consequences, including diarrhea in some patients, but not all. And we don't really understand the science behind most of these. And I think that's something that's going to require study for a period of time. Then there's heart involvement, there's cardiac advantage, and I think you all have probably heard recently about these unrecognized these strokes that are occurring in people that weren't even very sick, and particularly in the young. Trying to understand that, this outcome called COVID toes, where it looks like toes have been frostbit, skin rashes that people are getting, they're probably all related to this coagulopathy, this clotting dysfunction that we're beginning to see more and more in patients that have this. And I think that's also something that requires a lot of further investigation. So will there be a post-COVID-19 world? And I think the answer to that is no. I think we're in for the long haul with this particular virus. But I think focusing on the tools that we need to effectively adapt to a world where COVID-19 is circulating is where I think most of us believe we're going over the next few years, is really refining those tools, just as we've learned to refine tools for HIV. So I'll stop at that, and I'm happy to take questions.
Thank you so much, Dr. Corley. And I see some questions coming in the chat, but I'm just going to fire off a few, though, because I had some. So what is the pace of the discoveries like in terms of prior outbreaks? Is the science moving faster than, because it seems like things are moving fast, even though every day is very slow. But I mean, how is the pacing of what you guys are learning compared to previous viral outbreaks?
Well, I think there are two parts to that. The first is that people are putting out everything that they've discovered far faster than they've ever done before. Scientists and clinicians are usually pretty methodical and are careful about the type of information that they put out into the public. There are these preprint publication sites now that every day new publications are being put out. They're not peer reviewed, so that means we have to kind of separate the wheat from the chaff when we're looking at this stuff. But the amount of information that's out there daily is absolutely unbelievable. And I think this helps contribute to how we can move the science forward faster. But the other thing is that in previous outbreaks, people start repurposing. What did we learn? So for example, Moderna is using a vaccine that had already gone through clinical trials, not the specific RNA that they're using, but the type of methodology for doing an RNA vaccine had already gone through clinical trials to prove that it's safe to use in humans. So they were actually not required to they were not required to go through the animal studies before they took it out into the human population. The other thing that we do is everyone is going through the whole list of compounds that have already been approved by the FDA as being safe. And so that's one of the things that Rob Davey is doing with the Broad Institute, is getting their entire collection of FDA-approved drugs and running thousands of those through his assays to try to figure out which of those are actually effective. You've heard about remdesivir. That's a compound that was created by Gilead for fighting Ebola, but it's been repurposed to use in SARS-CoV-2. Clearly, it's not a cure, but it definitely seems to have an impact on people that are not seriously ill in terms of getting them out of the hospital faster. But knowing that, I suspect that they'll also be modifying that compound. So the answer is yes, things are moving faster, much faster. But they're still painfully slow, because until you have an outbreak like this and many, many thousands of people are infected, you don't begin to put a picture together of what the disease course is and what areas you really need to be working on. From a science perspective, I can tell you people here are just, feel like everything's going too slowly.
Yeah. Like all these other symptoms that are coming out, the COVID toes, the clotting issues, how unusual is this for a coronavirus?
Well, there are only 7 human coronaviruses. None of the others actually do this that we know of. There are large numbers of people that have had colds, and people that have colds generally they're not monitored for which virus has given the cold. Rhinovirus is the most common. The two coronaviruses cause colds. So we don't really know that the SARS and the MERS viruses don't seem to do this. So this seems to be very unusual. But I suspect people will go to coronaviruses that cause disease in other animals now and maybe use those as models to try to figure this out.
Rachel, I think we should probably handle some of these questions in the chat.
Yeah.
Do you want to take over for those?
Absolutely, because we definitely got a couple coming in. So one question, is there you just mentioned a publication site. Is there a centralized kind of clearinghouse of information where things are being shared efficiently that the public can access?
Many of the sites are, in fact, easily looked at by the public. So the bioarchive is a publication site that people put preprints onto.
Okay.
It's BioRIVX is the site, and if you just type that in, you'll come to it, and you'll be amazed at how many thousands of papers are already there.
Okay.
Lancet, the Lancet journal out of the UK, has a fabulous COVID-19 site of publications and information that is open to the public. They've made it free for everyone. The New York Times appears to have a huge number of people sifting through data. And I had a conversation with a colleague in the Center for Regenerative Medicine a couple of days ago about sometimes we found out about science that's coming out in the New York Times faster than we actually see it in a journal. And I wake up early in the morning, in fact, and I peruse through the articles in the New York Times just to see what they're talking about and what I do and don't know. To be honest, it's a fire hose that everybody's drinking from.
Yeah. Okay. That's interesting. But the New York Times, I wonder what they know. So another question. So you mentioned that a lot of groups are trying to use drugs that are already FDA approved, right, to treat. Everyone's moving forward as quickly as possible, even though it's slow. I'm guessing the FDA is probably really torn about how quickly to say things are working versus following the process. Should we be concerned that there's going to be premature approvals on anything, or you think that they're really trying to keep their heads on with this?
I think unfortunately the pandemic has taken on political overtones.
Mm-hmm.
And I think pressure is being placed where you don't want pressure to be placed. I know that the idea of allowing remdesivir to be used on an emergency use authorization, while they're still in the phase 3 clinical trials for that, was done thoughtfully. because they also had seen no harm. And I think the FDA is still going to require that. Some of the other compounds that have been tested, the chloroquines and hydroxychloroquines, unfortunately do have significant side effects and have been caused some damage. So I think we do have to be careful about that. And the push for therapeutics is one that I think concerns everybody.
Yeah. Another question, do we know how many times the virus has mutated?
Well, this virus, likely, almost certainly came initially from either a bat or a pangolin, because they've isolated sequences from those two species of coronaviruses that are 99% identical to this one, far more identical to this virus than SARS, for example. Now, all of these viruses, all RNA viruses mutate over time. And it's one of the ways, because of those mutations, that we're able to actually track where infections came from. So, for example, when people went back and started looking at the cases in New York and started sequencing the virus, you could tell that that virus actually came through Europe, primarily Italy, rather than directly from China, because of some mutations that had appeared during the course of infection. Now, do we know whether or not those mutations change the course of the disease? And I don't think we know the answer to that yet.
Okay, and we probably don't know whether that changes treatments and tools and diagnostics. We just don't.
Everybody and everybody is. I mean, luckily the spike protein has to maintain some semblance of similarity throughout evolution, because otherwise it won't bind its receptor. The polymerase, which we know, and the nucleoprotein we know evolve a little bit, but not much, because it'll kill the virus. So I think if it evolves over time, we may see it becoming a little more less virulent over time than currently, but we don't.
Okay, so we'll do one more question, and then we'll say thank you so much, because this isn't, I could probably ask questions for hours, and that probably wouldn't be helpful for right now. But what's the best way to sift through all of the information that's out there, right? I mean, you're looking at New York Times, other people, there's publications, there's pre-peer reviewed, post-peer, like what should we actually be doing to understand what's going on?
Personally, I don't think anybody can stay up with everything that's coming out. And so what we do is we kind of get together in various groups a few times a week, like the Mass Consortium for Pathogen Readiness. We have call-ins on a once-a-week basis of the 6 different interest groups. And because we've got a variety of different people who are interested in different things, they not only talk about what they're doing, but talk about what they've seen that's interesting. That's the way I tend to keep up, because I can't otherwise do it, and no scientist can. And so I don't think there's any one right way of how you do it. But for sure, get your news from reliable sources.
Okay. Brian, did you have one more question?
I did. I just wanted to touch on your last point about there is no post-COVID-19 world. I frame that a little bit more for us. I mean, is this something we're going to be dealing with for the rest of this generation's lives? I mean, are we going to, do you have any more thoughts on that, just to kind of?
Yeah, the reason I say that is because I certainly don't see it going away in the next year. You've probably heard Tony Fauci of NIAID say that he thinks this is going to become seasonal, just like the other 4 human coronaviruses that are seasonal. And that might very well be the case, and I suspect that all of us think it will be. I think we will continue to circulate this virus within the population for a while, simply because as we start opening things up, we're going to have small outbreaks and people are going to have to kind of clamp down a little bit in those areas, identify, trace. The more we have better diagnostics and we can do it routinely, the better off we're going to be. But I think there's always going to be small numbers of people that are probably carrying the virus that we're unaware of, that'll probably circulate it for a while. It depends so much on what they call herd immunity, which is when there's enough people in the population that will keep the population from widely disseminating the disease. For a virus like SARS-CoV-2, COVID-19, that means about 70% of the population will have to be immune, which is still an open question of whether or not having recovered gives you long-term immunity or not. So I think it's going to be a while, for whether it's forever or for a generation or for a few years, I don't know.
Yeah. A while where we're wearing masks and doing that sort of thing, or you think a while where we're still sitting in our...
While we're wearing masks and trying to social distance. It's been really interesting here in the NEIDL where virtually everybody's been working to try to spread people out and remind everyone to pay attention to social distancing at the very least.
Well, if you guys don't have a handle on that, I don't know who would. I just want to thank you. I can't imagine what this must be like for your staff. You guys, you know, having known what you were seeing coming down the road, I can't imagine the amount of stress, agita that was going on. I mean, I just want to commend you guys for all the work that you're doing.
Well, thank you very much, but I'll have to say that I think the stress level for me is much less than for people that are home, because I think, you know, I come to work, you know, I walk to work and I walk home. Yeah. So at least I have that break in the day. My wife is just climbing the walls because, you know, she can't. So I think people who are stuck at home probably have a much higher stress level than we do, to be honest.
Well put. Thank you so much for your words, and we'll have you back hopefully in a few more weeks, maybe with some really good news or better news or some progress.
Well, thank you.
Awesome. Thank you so much. So at this point, we're going to welcome Rick Clooney from Brainy IRB to chat. I asked Rick to give us an update on how Brainy IRB is working with some of their clients, right? We've had a talk in the past few weeks about clinical trials shutting down and things that are just becoming incredibly difficult. And if you had it in your plans, it's not in the plans anymore. So I thought it'd be great to have an update from Rick on how people can still be understanding what the scenario is, what it's going to be like moving forward, and what you can be doing now. So Rick, I will allow you to take it away.
All right. Thank you, Rachel. And thank you, Dr. Corley. That was sobering, but very informative. So, all right. If I can navigate this. All right. Well, thank you, Rachel and Mass Medic and Brian, for asking me to participate today. I appreciate that. We are an independent institutional review board, or IRB, known for short. And those of you who don't know what that means or what we do is we're an ethic review committee that is chartered with reviewing potential new research studies, and our charter is to protect the safety and welfare of the human subjects involved in these trials. We're independent, we're commercial, we're outside of the teaching hospitals and academia. and where, so we see a number of private studies as well as federally funded studies. One thing I wanted to center on today is how things have changed with our existence. In the last several weeks, we've processed 23 studies directly associated with COVID-19, and there's more in the pipeline. So we're just one IRB here in the country. And so as I think Dr. Corley alluded to, the activity is frenetic. And so we're part of that. And where our challenge is to help these new studies get on their way and still uphold the standards of all IRBs, which is that we all prescribe to, that, and that is to enforce this, the safety measures that are have to be enforced in order for human subjects not to be exposed unduly to undue risk. So we typically run 2 full IRB meetings a week. At this point, we are running ad hoc meetings on an as-needed basis. We have a all hands on deck type of mentality right now. Luckily, we have a staff that has been reviewing studies and preparing studies for our IRB for over 22 years. There's a great level of expertise there. There's a huge amount of collaboration with these new studies and with the researchers that are submitting them. And we're all on high alert, as easy enough to understand, to process these studies, to get them, get the right questions asked, get the answers to those questions from the researchers so that they can be approved and that they can get back in the hands of the researchers and for this critically important research to be conducted. The types of studies we're seeing, the new studies they range from drug and device trials to observational studies, statistical analyses, data repositories. It runs the gamut. What we're doing, Brainy, is besides accelerating our response to everything manyfold, is we're reducing our fees, or in some cases, eliminating fees for the COVID-19 related protocols. Our turnaround times are, in some cases, same day as far as a completed submission. We get it in front of the board. Some of these can be expedited depending upon the complexity of the study. So those are some of the things that we've got going that are unusual or extraordinary. We've always run, well, in the last 15 years, we've run a virtual meeting, so that has not changed. We were somewhat prepared for this, for working under these conditions. We have electronic submissions, which streamline the whole process that researchers can have access to, and their questions are asked. There's tutorials along the way. And as I mentioned already, our staff is very experienced and very holds a great level of expertise. So those elements all help in getting this research through the process safely. Our whole charter is to weigh the benefit to society versus the risk to the individual human subject. So that's what it all boils down to. Let's see. Whoop. Okay, so there's two types of things, and Rachel alluded to the second. There's new studies, but there's also existing studies, and we've seen it's not hard to comprehend when you're running a study that involves an intervention that involves human subjects interacting with researchers. Everything is grinding to a halt under these conditions. So, and that affects a number of things. A lot of these protocols are falling out of compliance. And so it requires collaboration with the IRB as far as amending the studies so that they can somehow get back into compliance, that they can continue. What type of conditions there might be when they resume the studies, what constitutes reportable deviations, what doesn't. So there's questions upon questions. Do I need to amend my protocol? Do I need to reconsent subjects once we get back up and running? And the answer to these questions in every case is it depends. And we take it on a case-by-case basis because every study is different. But it has put a lot of stress on the research community, as you can imagine, and we're trying as hard as we can to be there to collaborate with them to make these changes, and obviously with the overriding charter to protect our subjects. All right, let's see. The Brainy has a number of various arms. We own the city program, which is the leading provider of research and education. We have programs where we can help these new researchers build protocols. We have a bioethics committee, as well as a social behavioral educational committee, as well as the normal IRB. And we have other services that we're prepared to help people with. So I guess I'll stop there and see if people have questions. Yeah.
Rick, this is Brian Johnson. So have you seen in the non-COVID related studies, has there been a dramatic slowdown of new studies that you've seen?
There has. People are, there's various elements involved. Some of them are just the logistics of how they're going to carry out a study without interacting with subjects. There's the economics of it. Does this still make sense as it did prior to the pandemic onset? So there has been a stall in new studies, and a lot more of the COVID-19 studies are getting researchers' attention.
I'm curious about the, in a case like this where we have a global pandemic and on these studies, I mean, you talked a little about sort of weighing the sort of ethics and does any of the calculus change in terms of the protocols of the study when you have a global event like this? Or is that up to you guys to maintain those standards?
I mean, I think the answer is yes, it's definitely up to us to maintain those ethics. I think, human nature is to embrace the urgency, and but we need, as an organization, as an institution, IRBs in general, we need to maintain those standards. And so that's the challenge.
Yeah. Has there been a comparable period like this in your recent history? I mean, I know we're in a unique situation, but in terms of the flow of new studies that you're seeing, have you seen a
No
variable slowdown? Okay.
No. I've never experienced it. I've been doing this for 18 years. And no, I've never seen anything on this scale. So fast, so soon, so, impactful.
Yeah.
So, yeah.
Just a quick question. I do have one. Is there any advice you would give to companies who are looking to submit something for non-COVID related trials? Do they plan for that now? Would you recommend a hybrid? Do you plan for when things, go back to quote unquote normal? Any kind of tips you've been giving out to people in that regard?
Yeah, it's a good question. The advice I give to people is to collaborate with us. Our staff, as I've mentioned, has been doing this for a long time, and we have a lot of experience. We know what the IRB is looking for, what their concerns are, and we can oftentimes help researchers in designing a study that is not only achieves what they're trying to do as far as efficacy and showing results, but it also is safe for the participants involved. So my advice is, don't hesitate, reach out and involve us in your designs, and we're there to help.
Perfect. I think that's a great way to say thank you. And if anyone has any more questions, we do have another minute. Please pop them into the chat bot. But other than that, I think that's really great. Rick, really do appreciate you giving us kind of an update on that. I think it's really interesting how things are shifting. And planning for that is a really important thing. And it's really crucial, I think, for companies. Companies still hope to be companies once this crisis has, reached its next stage and its next phase. And we have to consistently be thinking future and forward, and how we can make ourselves make best use of the time and be in a position to be successful after. So I think this is really helpful.
Oh, good. Well,
So
thank you for the opportunity.
absolutely. And I guess on that note, we will say thank you to everyone for joining today. As always, video of this will be made available. We'll send it out to everyone in our newsletter next week. If you're not on the newsletter, please email me at rachel@massmedic.com, or you can go to the website to sign up. But we will get you all the information. I think it was a really great program, and big thanks to Dr. Ronald Corley and Rick Clooney from Brainy. And everyone have a wonderful day. I hope to see all of you tonight at the women's event at 5 o'clock. All right. Talk to everyone soon.
Wouldn't miss it. Thank you, everybody.
Thank you.